right when you came back from your sabbatical, he just came with the idea that we have so much archaeological, archaeological, and evidence in Israel, and he went to Professor Patricia Smith and said, why shouldn't we start ancient DNA in Israel? So I think the father of ancient DNA in Israel is...
Now I'm going to tell the real story. I want to thank Gila and Liran, but I have no excuse. You know, you guys are working on ancient DNA. I am ancient DNA. And I invited a professor who was my age a couple of years ago, and he came and he said, I'd like to tell you that I'm glad to be here. Of course, I'm glad to be anywhere. So, you know, that's how it goes. Okay, what I'm going to do is I'm going to tell how the story began. And again, when you get old, you can do whatever you want, so I might as well tell you. The thing actually began in 1991 when a guy named Joe Zayas, who I hoped would come, he was a curator at the Rockefeller Museum, and he said, can you get DNA out of a mummy? The next morning, the first Gulf War broke out, and I really couldn't think about mummies, but by chance, I met Professor Patricia Smith, who I had hoped would come too. She's now Pat for the rest of the talk, in the coffee shop, and I said, what archaeological material do you have consisting of 100 individuals? I'd come from public health, and I thought, you know, singular mummies didn't interest me, populations did. And she answered immediately, the Ashkelon Babies. And I'll come back to them and tell you about them. They were absolutely critical. We needed molecular biology. I knew how to extract DNA, but that was about the limits. And Ariel Oppenheim, and I don't know if Filon is Dory here now? No, okay. She came to us with Dory Filon, and with that, Marina Feyerman arrived in the country. We began on the baby sex, we began on thalassemia, and we began on TB because Marina had a friend from Lithuania with 5,000 skeletons. Many of them, I think 1 in 80, showed morphological signs of tuberculosis. So we began. Then Gila came. She asked the same Josias, the same curator, come on, where are the dog collections? There are dog cemeteries here. And he said, oh, come on, forget the dogs. We have 10,000 pieces of ruminants, the Dead Sea Scrolls. Now, during this period, the first question was, what does tanning do to DNA? And during this period, we had to hide our shoes because Gila was running around snipping pieces of leather out of our shoes. You know, it was a difficult period. Well, then, Alma joined us, and you've all heard Alma this morning. And she said, come on, this ancient DNA stuff is frustrating as hell. I'm going to use modern DNA and do Y chromosome. And after hearing all this talk, you can actually infer something from modern DNA, you know, a little bit. Then Gila discovers Mark Spiegelman, who's sitting in the front row, digging in Beit Sha'an. She brings him to lunch, I think in Beit Belgia. I think we met there. And this brought in one of the strangest individuals, a guy named Bruce Rothschild, and I'm going to show you a cartoon about Bruce in a minute. But it was Mark who brought us together with Helen Donahue, and that was critical for many, many of the things that happened here. Hillel Bercovier introduced us to Spoligotyping, and that was also an exceedingly important breakthrough for us. Jan Guneveg was a guy working with neutron activation, and he kept saying, come on, let's do Qumran without the scrolls. There's just so much there, the pottery and the fibers and all kinds of things that we could look at, and we began on that. Benny Klein was a physician who did part of his training in Italy, and he's not here, so I can say he's sort of a wino, but at any rate, he got us interested in grapes, and we were upstaged this morning by the talk by Drury, but I'll come back to that. Israel Hershkovitz, who will come tomorrow, got us involved in Atlit Yam, where we had people living with cows, and so we could ask questions about zoonotic transmission. Then a man named Shimon Gibson dragged in an old rag, which turned out to be a shroud from the Second Temple period, the only authentic shroud of the Second Temple period. The Turin shroud is probably a pious fraud. And then Suar Arakat, who will be here tomorrow, when waiting to get better bones from Jericho, began on modern DNA of TB on the West Bank and found, unfortunately, that there's lots of bovine tuberculosis on the West Bank. Oh, something's supposed to happen. This is Bruce Rothschild, and I just want to tell about him. He brought us something called an extinct bison. He's a rheumatologist. He was in then Youngstown, Ohio, my hometown, and my sister went to him as a patient. And she said, I think this guy is more interested in dinosaurs than in people. Because all over his office there were bones and funny things. A funny guy. And this is a cartoon from a book that I'll tell you about in just a minute. So there were some basic considerations. We really had to understand the archaeological context. And Gila is right. I didn't say we had all this archaeology. I didn't even know there was archaeology. I knew that we had some ancient DNA. I really began there. But at the same time, I really didn't believe that the ancient DNA was going to tell us the whole story. It had to make sense phylogenetically. We had to do microscopy. We did lipid analysis, spectroscopy, neutron activation, scanning electron. All of these were absolutely essential to really put things into some kind of structure. And then Nebel really took us to robust modern DNA. You know, it was the major contaminant, and this has come up again and again, even in this period. It was our enemy in a way, and yet it had wonderful stories to tell, and we had to learn how to deal with it. Well, we put out a book, and I left 6 or 7 copies in the side room. I think it's beautiful. It has 2 articles in it which make it worthwhile to carry this heavy thing home. One is by Pat Smith again on the whole structure of the Levant, but scroll forward from what we heard from Nama and up to the period of, say, 10,000, 12,000 years ago. It's a beautiful article. But there's also an article by Geoffrey Eglinton, who was the father of the Ancient Biomolecule Initiative in the UK. He's a deep organic geochemist or something like that. And it's a gorgeous article on any of the biological remains that you can find when you start doing archaeology. So if for some of you, if you want the book, if you don't want to carry it, write your name and address on a piece of paper and give it to me today or tomorrow. I think it's really worthwhile. Well then, in Pat's article, there's this statement, and I love it. The archaeological record does provide some indication of habitats utilized, diet, and methods of food preparation, as well as estimates of group size and interaction. The extent to which all of the above may affect the risk of exposure to different diseases in past and in their role in the evolution of modern population differences in disease susceptibility, etc., etc. This really was horrendously important for me to begin to understand this. Her subtitles in the chapter included paleodemography and settlement patterns, paleoclimate, plant utilization, animal resources, skeletal evidence for disease, and dental pathology. It's just a wonderful, wonderful chapter, and I hope you can see it. Another thing that really influenced me tremendously was a short quote from a book by Richard Pollock called Signs of Life. And I must tell you that now much of my work is in polymers, so this really speaks to me. Many molecular biologists are becoming historians. Little by little, they start to approach genomes, not so much as polymers, but as old manuscripts which document the history of life on our planet. As they learn more about old genes, they open a door to unexpected historical events that might later reflect on our ideas of molecular biology itself. And later, when I talk about tuberculosis, I'm going to talk about one of the insights into molecular biology. I admit it came to me late, but on the other hand, I think it was very important. And I think there are many things like this. With Mark Palin, he spoke of the fact that looking at ancient tuberculosis made him think much more seriously about modern tuberculosis. And I think there are lots of these spin-offs which are really very important. So we had to be multidisciplinary. The ancient DNA was important, but physical anthropology and ecology and evolution and systematics, all of these things were important, and paleobotany as well. So we had to go that way. Now, for the foreigners, you may not know, there is a kind of tension in Israeli archaeology between the maximalists and the minimalists. The maximalists really say that we're going to find in the biblical narrative the hints for the things we should look for. The minimalists say, well, let's do the archaeology and then just see, did the Jews really wander through Sinai for 40 years, etc.? And there's lots of feeling about this. And our feeling, of course, we're bottoms up. But what has interested me tremendously is the spin-off. And for example, when we talk about TB and leprosy together, we have something to say about the demise of leprosy. We also introduce new methodologies into the public health system in detection of tuberculosis. This young woman, Suhar, tomorrow, who's going to talk about TB, while waiting for good bones, she really discovered a focus of bovine tuberculosis in the West Bank. Gila prevented the introduction of what was just simply a feral goat into the nature reserves. So for me, this spin-off is as important as any. I'm just a dumb physician and not going to add very, very much to possibly historical understanding, but at any rate. But some of the questions which I'm going to talk about for the next few minutes are: where did the Israelites come from? What about Jewish priests? What were the diseases which afflicted the Israelites? Was human TB a zoonotic disease? And what can we say about the domestication of animals? On the origin of the Jews, I just have to quote Almut. And she tells us that from the Y chromosome, we really find that the Sephardim and the Kurdish Jews were genetically indistinguishable, slightly different from the Ashkenazim. And these were ancient genetic patterns. On the Jewish genes and social class, the Jewish priests, I have to quote Thomas and Bradman, but also a man named Skorecki, who's a clinician in Haifa, who collected buccal swabs from anybody who had a name like Cohen, Kahan, Sacher, Sacerdoti. These are people who feel that they're descended from the Jewish priests, and they really remain this way. The Levis do not share a group of about 6 genes, which is called the Cohen modal haplotype, and it's at a high level in the Kurdish Jews, less in the Ashkenazim, high in the Sephardim, and drops away in the Muslim Kurds and the Palestinian Arabs. And the cluster can be extrapolated back to maybe 3,000 years, so it does really fit part of the biblical narrative. More than that, the biblical narrative would say that we came from somewhere up in here, in Kurdistan someplace, come down, then become slaves in Egypt, come back. On that we can't say anything. But on the original origin, again, Almut said that we were like this. And in the world tree, the Jews are in this central cluster. So again, the feeling, the minimalists would say that we're just Canaanites. The maximalists would say that follow the biblical narrative. And it's interesting, I think. On settlement, we want to talk about animals and plants. And I'll start first with animals by going directly to Qumran, where the Dead Sea Scrolls came from, and where Josiah said there are 10,000 pieces. And Judaism was in turmoil 2,000 years ago. Any of you who have seen Monty Python movies know that everybody was crazy. And in Qumran, this really awesome site, some 800 different manuscripts were found. And to this day, people are arguing whether it's a library, a pot factory, a Roman villa, whatever, or were they really written there? And the question that Gila and I have asked, and Dan Bradley, I hope, will help us answer, were these flocks. And we have some 400 bones from the plateau in Qumran. Are they the source of the parchment, which would probably indicate the scrolls probably really were written there? Well, the site looks something like this. It's really quite awesome. And the pieces look something like this. In the light of the September 11th, when the 3,000 samples of human remains were robotized and automated, et cetera, et cetera, et cetera, we really thought of matching the pieces. But it was clear we didn't have the money or the manpower or anything. But instead, at a very recent workshop with John Guneveg, the National Archives of Holland and Google said, come on, don't do it that way. Do it by digitalizing the whole collection and then looking at the morphology, the scribal behavior, the color, the grain, etc. And Google is now deep into digitalizing the entire collection of these pieces. And I'm sure that much more text is going to be emerging. Okay, Gila did do the phylogeny. The scrolls do fall into an area with 2 primitive breeds, the Grimi and the Baladi. And again, all of the out of Israel strains fall into other clades. So again, we were able to make some headway. Again, we would love to be able to match the parchment to the bones, and for this we really need next generation sequencing. Now, in plant domestication, 2 of the most important exports were not wine and olives during biblical and Talmudic time. They were balsam and dates, strangely. In balsam, a guy named David Iluz at Bar Ilan University has brought in cuttings, and it's growing beautifully in Ein Gedi, and it's apparently very special to the climate. He brought a plant to the lab last week. We all smelled it. It's wonderful. It's apparently very labor intensive to harvest this stuff, so I don't know whether we'll recreate this. It was truly an industry in ancient Israel. On dates, a woman named Elaine Soloway, a great botanist, proceeded to get 1 out of 3 seeds to grow. Now there are thousands of these seeds around biblical archaeological sites, and I've failed 3 or 4 times to either get the date export companies or the JNF, the Jewish National Fund, to even think of gathering these and really doing a much more serious study. In the Talmud, the date is described, the Israeli date, is something like 10 centimeters long, disease resistant, growing in the worst salty soil, etc. And it would be really quite wonderful if we could recreate these things for their resistance genes and such. On the grapes, we were totally upstaged this morning by Drury's talk, and I'll have to meet them over coffee tomorrow and talk about this. All that we were capable of doing, I have to come back to this, I'll go ahead, was to really do microsatellites. We had this gene bank from the same Sataf that was described this morning, and Gila made us do microsatellites. We were doing brute sequencing, actually, and she stopped us. We had a lovely data bank of the entire Mediterranean, and when we really looked at the Israeli grapes, they came up with the Greek grapes. And this really fits, again, the narrative that the sea people brought their grape culture, their wine culture into the ancient Holy Land. So I'll go back for 1 second. Steve Weiner and Rivka Elbaum did a lovely study on, again, what's a good seed to extract. They found that they could evaluate the lignin by IR spectroscopy. They also did real-time PCR to determine the number of templates, and they were able to get some sequence, very minimal stuff, from olives. And olive is great big seed. Grapeseed is a teeny little fragile seed. But in fact, a third study by a man named Menon on grape seeds indicates you can do somewhat better than what we did with a few dead seeds, getting a very few microsatellite things. But these studies, which I think really would be significant, really cost money and are difficult. The Drury group at Ariel University are really working on this with deep sequencing, and I hope that we'll learn much more about this area. Okay, now just something about diseases. We really only have to talk about tuberculosis, leprosy, leishmaniasis, and malaria. And unfortunately, Marina has devoted real time to a very sensitive, species-specific test on malaria using archival slide material. When applied to 300-year-old material, the findings we won't talk about. We have one funny kind of malaria that is really now cited as an emerging malaria from monkeys, and we just don't feel it fits. In leishmaniasis, we have one case, very, very ancient, but it does fit the general feeling that we've gotten from the work, which our name is on the paper too, with Albert Zink and Nerlich, Andreas Nerlich, that again from Sudan, leishmaniasis passed in the Middle Kingdom, I think, to Egypt, so that we fit there. In leprosy, we have 8 Holy Land cases, 4 of which are juvenile. One is very, very ancient. And I hope, again, that Ben will light up on this and help us with this. On tuberculosis, again, we don't have many cases, but we have 2 very, very informative sites. One is Atlit Yam, and the archaeologist here is Hershkovitz, who will be here tomorrow. It's an underwater site. The bones are lovely and well preserved. They were under clay as well as under the sea. People lived here with cows. It's probably the earliest confirmed TB in human populations with morphological findings. And when done by Spoligotyping, it was M. tuberculosis and not M. bovis. Here is, I don't know if this is Hershkovitz, but he gave me permission to use the slide. And again, the library of animals were goats and cows and gazelles etc. And these were living with people some 8,000 to 9,000 years ago. And yet the tuberculosis is human tuberculosis. And before this, the common idea was that, again, it was a transmission of bovine tuberculosis to human. What we found both in the very ancient bison and here was that it can't be that way. There are major deletions in the bovis, which means that they probably separated long ago. Well, we had to have a good method, and what we used was called spoliotyping. And when I said earlier, in quoting Pollock, that again, sometimes we really learn very, very special things. The rage now in molecular biology are CRISPRs. Everybody is using them. They're very strange. Here you can read clustered, regularly interspaced, short, palindromic repeats. They require very special spacers with direct repeats between them. And it turns out that this is a prokaryotic immune system, which is a kind of record of the plasmids and phages which invaded the cell, and it can act against them very much like in eukaryotic cells, inhibitory RNA works. Well, it turns out that in Mycobacterium tuberculosis, there is this area which we used, and what you do is you get a gel, and you label one of the primers, and this is robust modern DNA. Bovis is always missing these last 5 spacers. Human always has them. This is a gamish of all kinds of ancient samples, and you can see it's a horrible thing to treat statistically, but on the other hand, always there are representatives of the last 5 spacers. And so there's really very, very little evidence of bovis from archaeological samples. And as Gila said at lunch, if you find a cow that's infected, you get rid of him immediately. So it's even hard to get modern bovis samples from the vets. They don't like to give them out. Okay, more than that, there's validation from lipid analysis. And this is the work of Minikin. And he did for us, or with us, both the Atlit gem material and the ancient bison. And it's exceedingly definitive, good stuff. The mycolic acids are species specific, and I think that's very, very interesting. And I'm getting to the end. About 2,000 years ago, the Israelis know where the Cinematheque is. Below it, there's a site that looks like this. The archaeologists knew that it had been robbed very recently because they found outside of it lots of broken ossuaries. The burial habit was to put somebody in a niche, and a year later you take out the bones and put them in these boxes. But they knew that in this tomb there was an unopened locus. And so inside, this is the entrance area, and here, something like that. And there was this one guy in a closed loculus. And in the back, when they got into this, there was this black rag. Well, they immediately thought of this, that it was a burial shroud, and it truly was. It was a fiber with a simple over and under weave. It was wool, as seen by the scanning electron microscopy. Probably from what we understand from the great specialists in the Antiquities Authority, it was probably an imported fabric, even at that time. The guy's hair was clipped very nicely and with no lice. He was upper class, and it certainly was an authentic shroud, unlike the Turin shroud. Again, there were bones. One was very, very strange. These are all the bones from the first loculus. He had 6 positive bones, probably meaning that he had miliary TB, and that's what killed him. 2 of the babies were positive, one at 2 sites and one at 1 site somewhere there. And we know nothing about their death. But we knew leprosy was rampant in the old city. And at the very same time, Helen Donahue had developed good primers for leprosy. It turned out then the guy with the 6 positive bones for tuberculosis also had leprosy. And the punchline here is he was infected with both leprosy and TB. We had a collection of about 32 samples that were either positive morphologically or positive for tuberculosis or leprosy, and it turned out that 10 of these were co-infections. I think later Zink found a somewhat lesser number, but other co-infections have been found since. The contemporary literature reveals that tuberculosis is a common cause of death in leprosy. Leprosaria were a common feature in medieval cities. They disappear with the Industrial Revolution. We sort of think, and I think this will turn out to be right, that leprosy lowered the immune response and TB came in and gave the death blow. The Shroud individual probably was disfigured by leprosy and therefore his locus was unopened. Finally, just to end, the Ashkelon Babies. There were 100 baby parts found in a sewer near a bathhouse, which was certainly a brothel in late Roman times. It was probably in the red light district of Ashkelon. And the question that was asked, what was their sex? Because this would reflect on whether it was some kind of cultic death. These were really within 2 days of birth. And Marina adapted a forensic method of the amylogenin gene to shorter pieces. And I'll just show you in a second what she did. And this also took us together with the thalassemia into nuclear genes. And up to this time, we were struggling with pathogens, et cetera. So this was really an important study for us. There's a deletion in the X chromosome so that boys give 2 bands and girls give only 1, shorter band. And it's a very definitive test. What Marina found was that the 43 left femurs, 19 specimens were positive, 14 were males, 5 were females. And in her article, she really discusses Roman infanticide. And it was very clear there is information in letters saying, "Dear Matilda, if you have a boy, keep it. If it's a girl, discard it," etc. And again, the bathhouse being tied into prostitution and the brothel is apparently very, very clear in Roman literature. And so again, those of you interested in brothels and such things is a good place to look. Oop. So I think this is where we stand. We're way behind in next generation genomics. And again, this is why you're all here. This is to help us. And we have the machinery, but until Liran came, we couldn't yell at all about bioinformatics, but he's here now, so he's going to solve most of our problems. We will be doing, I think, more in the physical sciences because I really find myself in the nano lab much more than I ever thought I would be. And we have a wonderful facility very, very near here. Microbiomes really attracts us, and we've been thinking about this for a long time. We have no results yet, but I hope that we really will. And Mark made wonderful ties to Korea. So Mark and Gila, and tomorrow you're going to hear Danny Shuval, did the first viral genome with hepatitis B. I'm sorry, the second. The first was influenza, but this was the second that was done. And I think that we're going to be hopefully doing more. It's not easy. So these are what I hope we're going to do. Okay, thank you very, very much. And please remember the book. It's in the other room. If you want it, please.